So, hi everyone. Let me just put you guys like this just before I start. The idea of this presentation is both for me and Alexey to present a bit of the work that we have done together with the ERGA community to ingest these genomes and have them available, the work that is still going forward, and Alexey will show a bit of the ERGA data portal. So I'm sure most of you are already very familiar with the European Nucleotide Archive, so I'm going to present a few slides just for context, and then I'll move forward for the main thing. So the ENA is one of the sequence repositories, part of the INSDC, the International Nucleotide Sequence Database Collaboration, together with NCBI and DDBJ. We are a global core biodata resource and an Alexey core data resource, and we hold a comprehensive record of the sequences, the DNA sequences. We have a broad scope going from raw data to assemblies and annotations. We're connected with other resources, and we have a number of tools that allow you to submit, retrieve your data, and so forth. In terms of our data model, this is organized into 3 tiers, which includes the reads, the assemblies, and the annotation. And all the data is enriched with additional metadata and connected with samples and taxa. Currently, assemblies and annotation are paired. And I'll go into the currently in the latest parts of my presentation. You're probably all familiar with the ENA metadata model, where we have the study that joins everything together. We have the sample information, where the organisms or environmental samples, if we're talking about metagenomes, are described. We have the experiment and runs, where the raw reads are described. This includes the data and the metadata in the experiments, and the data analysis, where that includes, for example, genome assemblies, annotations, but also other types of interpretations of sequence data. So what has been going on together with ERGA, and how do we present this data towards the global users? So initially, when these projects started, we noticed that we needed a little bit more complex structure in terms of projects, so that we can work together as a community and have the users understand that things are connected and that things all go within or beneath a different project. So we have a hierarchical bioproject structure, where you can see here the EBP as a top layer. And then you can see here the ERGA and the different projects that exist within the ERGA. This is not a comprehensive view. There's much more projects there. This is just a little snapshot with some examples. And you can see there's different layers of umbrella projects here. And then you have the data projects, when you can include the raw data, primary and alternate assemblies, or different haplotypes. But you can also include metagenomes, assemblies, cobionts assemblies. So you can put all of that beneath a species and beneath its different sub-project that is part of the ERGA initiative. And here is a display of how this is presented in our browser. One other thing that is very important for us, and that allows having rich contextual information around the data that we hold, is metadata, and we try to work together with the community around these metadata standards. So we have, in terms of sample, we have metadata checklists that are guided by sampling working groups and the community. In the case of ERGA, this has been totally driven by the community. This rich standardized metadata facilitates the linking with the source information. And we also have some general minimum requires, which are INSDC standards. That includes the spatial temporal information like country and collection date, organism taxonomy, which is linked with NCBI taxonomy. And we also have a missing value reporting standard that should be used when submitting this information. In terms of ERGA, we have the ERGA sample metadata manifest, and this was all community driven. This is based on the initial manifest that was developed by DTOL. And that translates into ENA in a Tree of Life checklist. So the Tree of Life checklist is not exactly the same as the ERGA manifest, because the ERGA manifest has some additional metadata, mostly for tracking and some specificities that are not included in the sample that is submitted to ENA. But both of these, both the manifests and the checklist, evolve together with the community as we move forward and the sequencing scales up. In terms of data submission and presentation, I'm going to talk about this example because this was an example where this is not so this is a detail example, which is within, it's also part of ERGA. And this was something that we had to work together with you, with the community to actually have this data in, because this was quite a challenge at the time. So this is the European mistletoe. This is a huge genome. As a used genome, it caused some issues, both in processing, which we had to improve at the time because of the size, but also because we do have a technical limitation, which is the sequences that have a maximum length. The largest chromosome has 100 gigabases, so it's much over the maximum length, as you can see here. So these chromosomes had to be split into different sequences, and we had to both work together with our colleagues in INSDC to define the standard, how these should be named and presented, and also improve our presentation in the browser so that a user can come here and download all of these sequences in one go. So this was an example where we had to work together to be able to get this data in and share it across INSDC. In terms of advanced search, we've also made some improvements to improve the access to these genomes. For example, you can go and search within all projects that are beneath the ERGA umbrella, and you can access all the, I think it's like 6,000 assemblies or over that you have under this umbrella. So you can query this and you can filter this out more, but this is just as an example of what you can do. One other thing that we have been working throughout the years is improving biodata linking between the data we hold at ENA and other data resources, which is valuable in terms of data reuse. So we have done this in the scope of a number of projects. So we link with other EMBL EBI resources, but we also link with external resources as Silva, as Unite. We link to data publications platforms, and we have recently built links both with GGBN, the Global Genome Biodiversity Network, which allows linking of the sequence data with the biobanks where the samples are kept, and with the BOLD data systems, allowing to link the sequences at ENA with the barcodes that are specifically submitted there and with other resources they have available. And this is just an example of our cross-reference search where you can pick the resource you are searching for. And from the link from the sessions there, you can link both to the resource that we're linking to and to our own records. And within our records, you can also link back to the resource and vice versa. So in this case, we have a bidirectional linking. So the last thing I wanted to bring up here as an information for now, because this is still in the development, is the efforts that we have been doing within the last years around decoupled annotation. So currently, assemblies and annotations are submitted coupled, so within an EMBL flat file. And what we're working towards is to decouple this and have an assembly submitted as a FASTA file and then the annotation as a GFF3. What this translates is as an annotation being a new analysis type. So you submit the assembly, just the sequence information, and then you can have different interpretations of the annotations within that assembly. So it allows a little bit more of community collaboration and having different views of an assembly annotation. These annotations will reference the assembly and its specific version, and it's independent of the assembly version. This is work still under development, but as Alice mentioned, it should be ready in 2026. So we're on the final stages of implementation, and we'll start testing this quite soon. So this is my final slide. So we are working with the community and making efforts to allow this biodiversity genomics at scale that we see as a general initiative. Currently, we have reference genomes for over 3,000 species, and this is already a month old, so I guess we have a bit more at the moment. We see increase of the volume of data downloads too on the user perspective, which also shows that the community is aware of these initiatives and it's actually looking to use the data. And we're basically trying to work to have fair reference genomes and enhance the use of this genomic data in biodiversity applications. And I thank you for your attention, and I'm going to pass the words to Alexey.
Hello, everyone. My name is Alexey. I'm working as a project lead at BI. My team is primarily responsible for ERGA data portal. Next slide, please. So apart from ENA, it's a slightly different portal. So it's still based primarily on ERGA umbrella ID, but we aggregate data and metadata from different sources, including ENA, biosamples, ensemble, NBN Atlas, and others, and just aggregate everything under unique tax ID. And here's the link for Biodiversity ERGA data portal. Next slide, please. So the main page looks like this. Next slide, please. So it's presented in the center. You see that the main table with pagination and sorting. On the left-hand side, you can see filters, and at the top of the table, you can see a search bar, and it's possible to do search for species of your interest using scientific and common names. Next slide, please. Then it is possible to make sure that your results even more clarified using a set of predefined filters on the left-hand side. So it's possible to filter by data status, symbionts, metagenomes available for your species of interest, experiment type, project type, a project name, and phylogeny. Next slide, please. Once users are satisfied with their results, it's possible to download the table as a CSV file. This is just the metadata. And the next slide, please. And if you would like to download data, there's another button, download data, that will redirect you to our Python downloader utility. This is just Python script with different sets of parameters, but basically you can download Docker image to your cluster to define what kind of species, what kind of data you need, and you can download the data that you need to your machine in parallel threads. Next slide, please. And as I mentioned, we aggregate different data and metadata across tax ID. So other external references include genome nodes available for this species, cross references with GORD, TOLQC, and NBN Atlas. Next slide, please. So every species name in the table, they're actually links. So here, if you click on the link, you will be redirected to the details page. Next slide, please. On the details page here at the top, you can see 5 tabs. So the first tab is metadata. Next slide, please. Basically, this is where we show all of the biosamples that are linked with this species, and you can filter these biosamples by sex, organism part, or the status. Next slide, please. So next up is the data itself. So here at the top, we are presenting annotations from Ensemble as well as a link to Ensemble browser. Down at the bottom, you can also download assemblies and raw data from ENA. Next slide, please. So next up is linked to scientific publication reporting the reference genome. This is just primarily genome nodes. Next slide, please. Then we have cross-references and some metadata from GORD, just to include here we're showing genome size and BOOSCA completeness, but we also have a link where you can to the GORD where you can read the full report about the species. And the next slide, please. And finally, last up, we have a sampling map just to show where the samples for the species were collected, as well as species occurrences map from NBN Atlas. Next slide, please. Another tab, so this was the data portal tab. Another tab is the status tracking tab. This is basically where you can check the status of your species of interest. So all species go through 4 different stages. First one is metadata submitted to biosamples, then raw data submitted to ENA, then assembly submitted to ENA, and final status for now is annotation complete, and I hope to see in 2026 annotations submitted to ENA. Right, next slide, please. Another tab is sampling map. This is where we show where samples were collected for all ERGA species. And you can use different tools within the map, like you can use lasso or just like a square box where you can select some geographic region on the map, or you can filter your species of interest by scientific common name, data status, or experiment type. Next slide, please. Another way to try to find your species of interest is to use phylogenetic tree. Again, at the top, it's just possible to do search by scientific or common name, or you can use the graph in the middle. So single click will just expand the current graph. This is current phylogenetic taxa. And if you want to filter to show all species from this specific phylogenetic group, you can just double click on a node and it will filter and show you all the species for this specific plate. Next slide, please. Yeah, this one. We also have some like visual dashboards. This is just additional way to filter and find the species of interest using different metadata fields and data fields. Next slide, please. Another tab is publication. This is where we aggregate and show all the genome nodes that were collected so far for ERGA species. And the next slide, please. And for those who'd like to have programmatic access, we have public open API, so you can just check the documentation and use our data portal without the need to use the UI, which is basically used from your terminal window. And the next slide, please. Yes, that's thing. Thank you very much. So we have a dedicated help desk. So if you have any questions or feedback, please just share your email. It's quite simple, biodiversity-portal@ebi.ac.uk. Thank you.